Invisible roads carry secrets across forests. We think trees talk only through light, yet they whisper in chemical code. They send volatile whispers, shuttling sugars and signals through soil pores like a hidden highway. How can a silent network outpace lightning? But how do these invisible currents survive the dark, damp earth? The secret is in fungal hyphae, tiny bridges that carry life's code. Welcome to the hidden world beneath our feet. Think of roots as cities and fungi as their roads. These fungal highways are made of microscopic threads that weave through soil, connecting trees across miles. They carry sugars, hormones, and tiny chemical messages like letters in a secret script. In this segment, we'll break down how these signals travel and why they matter for forest health. Fungal hyphae extend from a single root into the soil. They grow laterally, forming an extensive filament network. This network connects many tree roots across a forest landscape. Arbuscular fungi penetrate individual root cells to form small sacs. Mycorrhizal fungi coat the root surface, creating a fungal mantle. Both types exchange nutrients through these hyphal bridges. Carbon from photosynthesis travels down the hyphae to the fungus. In return, minerals like phosphorus move back toward the tree roots. The network functions as a communication highway between trees. Signals travel along hyphae via electrical and chemical cues. Pause 0.8 Fungal hyphae release a cloud of volatile organic compounds into the surrounding soil. These molecules are small, lightweight, and can move freely through microscopic pores. Diffusion is the main driver. Concentration gradients push VOCs from high to low areas. As the vapor travels, it encounters root hairs that have receptors tuned to specific signals. Binding of a VOC to a receptor triggers an electrical change in the root cell membrane. This signal is transmitted along the root network, coordinating growth and defense responses. The process repeats across many fungal root pairs, creating a distributed communication web. Because VOCs are volatile, they can travel several centimeters beyond the immediate hyphal zone. Soil moisture and temperature modulate diffusion speed, affecting how far signals reach. Thus, fungi use chemistry to link distant roots without physical contact. Pause 0.8 At the root-fungus junction, chemical dialogue begins with glutamate release. Glutamate binds to fungal receptors, triggering intracellular signaling cascades. In turn, fungi secrete chitin fragments that act as recognition cues. These fragments are detected by plant pattern recognition proteins on root cells. Binding initiates transcriptional changes that promote hyphal growth into the cortex. Conversely, plants produce trigalactones that attract fungal partners to the interface. The exchange is tightly regulated by feedback loops involving calcium fluxes. Calcium spikes reinforce receptor sensitivity and stabilize the symbiotic partnership. Thus, a bidirectional chemical handshake sustains nutrient flow between roots and fungi. Understanding these signals illuminates how ancient forests maintain ecological resilience. Pause 0.8 Glucose transporters sit on the hyphal membrane. They bind glucose released from fungal metabolism. Binding triggers a conformational change that opens the channel. Through this channel, glucose moves toward adjacent plant root cells. The movement creates a local concentration gradient. Root cells detect this gradient via their own transporters. Detection initiates signaling cascades that modulate nutrient exchange. Thus, glucose serves as both fuel and communication cue. The fungal network coordinates activity across the forest floor. This coordination supports resilience against environmental stress. Pause 0.8 Plants release desmonic acid and salicylic acid into the rhizosphere. These molecules diffuse through soil pores and reach fungal hyphae. Fungal receptors bind the hormones, triggering intracellular signaling cascades. Signal transduction leads to altered transcription of defense genes in the fungus. Conversely, fungi secrete effector proteins that modulate plant hormone synthesis. These effectors can inhibit jasmonic acid production, reducing plant defense costs. The balance of signals establishes a coordinated response between root and hyphae. When pathogens attack, The fungal partner amplifies salicylic acid signaling in the plant. This amplification primes neighboring cells for rapid defense activation. Thus, hormonal crosstalk functions as a bidirectional communication hub that optimizes resource allocation and protection. Pause 0.8 Soil moisture controls the diffusion of fungal signaling molecules. When water content rises, molecules travel farther through root networks. In dry conditions, signals become trapped in micro-pockets of air. pH levels alter the charge on signal compounds, affecting their stability. Acidic soils tend to degrade certain peptides more rapidly. Neutral or slightly alkaline environments preserve these signals longer. Temperature influences enzyme activity that synthesizes and breaks down messengers. Cooler temperatures slow production, reducing overall signal strength. Warmer climates accelerate synthesis, but also increase degradation rates. The balance of these factors determines how far a message can spread. Heat map overlays illustrate zones where signals persist versus fade. Understanding this modulation helps explain fungal communication across ancient forests. Pause 0.8 Early vascular plants first encountered soil microbes during terrestrial colonization. These interactions prompted the evolution of simple chemical signals. Signal molecules such as strigolactones guided fungal partners to roots. Fungi responded by secreting lipid oligosaccharides that activated plant receptors. This reciprocal exchange established a rudimentary communication network. Over millions of years, signaling pathways became more complex and specific. Modern mycorrhizal systems use diverse phytohormones to coordinate growth. Root exudates now include sugars, amino acids, and secondary metabolites. Hyphal networks integrate these signals into a coordinated forest-wide dialogue. Thus, the biochemical foundation of fungal communication traces back to early land plants. Pause 0.8 Researchers introduced a fluorescent dye into the soil near two saplings. The dye entered fungal hyphae that connected the roots of both trees. Over several hours, time-lapse cameras captured the dye's movement along the network. Each image revealed a bright filamentous trail tracing the path of the molecules. The speed of transport varied with temperature and fungal activity levels. Data showed that signals could travel several meters within a single day. This rapid exchange supports coordinated nutrient sharing between distant trees. It also suggests fungi act as an underground highway system. The study confirms biochemical communication is not limited to adjacent roots. Future work will map the full extent of these hidden conduits. Pause 0.8 Fungal communication operates through a complex web of chemical signals that travel along root systems. These signals include volatile organic compounds and soluble molecules that can be sensed by neighboring fungi and plants. When drought stress occurs, mycorrhizal networks transmit water-saving cues to distant trees. Similarly, nutrient-rich patches are marked by fungal exudates, guiding roots toward fertile zones. Pathogen threats trigger defensive signals that prime adjacent trees for resistance. By mapping these biochemical exchanges, scientists can identify which species act as hubs in the stronger network. Conservationists use this knowledge to select tree genotypes that enhance network connectivity. In restoration projects, planting mycorrhizal-friendly species boosts soil health and reduces erosion. The resulting resilience lowers the likelihood of large-scale forest decline during climate extremes. Thus, understanding fungal signaling informs practical strategies for sustaining healthy forests. Pause 0.8 Fungal hyphae form invisible highways that shuttle sugars, hormones, and volatile signals across forest roots, with transporters and environmental conditions shaping the flow key to forest resilience.